many of you are interested in, which is mitophagy and regulation of dysfunctional mitochondria and how that relates to disease. So, I think that many of you in the audience have seen movies like this of mitochondria. And really, movies like this is really how I fell in love with the cell biological aspects of mitochondria and really got interested in these questions of how organelles get spatially regulated in cells. Right? We know mitochondria move around, they divide, they fuse, they form these elaborate structures. And I think it's a fascinating question of how the cell knows where to place these organelles, what their size and shape means for their function, and whether a mitochondria out here might be different necessarily than a mitochondria that's kind of more in the perinuclear region. So I think I got my start by taking movies like this at a cell biological level, where you just take a fluorescent protein targeted to the organelle and watch over time. And one of the reasons that we're really interested in mitochondria in particular, as I ask these questions about spatial organization, is because, as we all know, they have two membranes, and the intermitochondrial membrane is also highly dynamic and highly spatially organized. So the cristae membrane, the inner membrane that forms these invaginations that house respiratory complexes, can look drastically different depending on the cell type that you look at, and that obviously very highly correlates with the different respiratory demands that different cells need. So you can look in a cardiac cell, for example, and they will have mitochondria that are just jam-packed with cristae. You can look in glycolytic cells like stem cells, they're very disorganized cristae. And so kind of from this internal perspective, we can also ask questions about how you organize these cristae membranes within the organelle. And then mitochondria are amazing because they have all these additional layers of complexity, right? So I think, beyond respiration, we can think of them as these metabolic organelles that have hundreds or thousands of different metabolic reactions that have to occur inside the organelle. They have other jobs like regulating cellular calcium homeostasis, mediating the apoptosis pathway, and then even layering on more complexity of spatial organization. They have their own genome that has to be organized within the cell. And as was alluded to in the introduction, they're contacting the ER and just about every other organ of the cell. So as someone who loves looking at beautiful structures under microscopes and thinking about the spatial organization, they're really the perfect organelle to study. And I love showing this movie from Stefan Jakobs' group because, now with latest and greatest techniques in super resolution microscopy, we can actually start to visualize these cristae membranes in real time. The red in this movie is the mitochondrial nucleoid structures. And you can think where we're going with the spatial organization. We can start to place all of these different processes in real time in the context of this intermembrane organization. And so the broad questions that we're really interested in asking in the lab are twofold. One, at a mechanistic level, how do you spatially and functionally organize the organelle? How do you control all these functional processes within this elaborate network? And then secondarily, depending on the metabolic needs of a cell or even a subcellular location within a cell, how do you dynamically modulate these structures and basically control all of these different functions? And so to ask these questions, what we do is we kind of look at the interplay between all of these different aspects of spatial organization. So we talked a little bit about how the cristae membranes can kind of dynamically reorganize depending on respiratory demand. We talked a little bit about the fission, the fusion, and the motility of the organelle. We're really interested in how these two things crosstalk with each other. We're also interested in how mitochondrial interactions with different organelles actually impacts cristae organization, impacts these dynamic behaviors. We're also really interested in how the metabolic needs of cells and the different functions of mitochondria feed into all these pathways. And so, as was mentioned in the introduction, we're really interested in the mitochondrial contact site and cristae organizing center complex. And the reason that we love MECOS so much is because we kind of think of it as this key to internal organization of mitochondria. So for those of you who don't think as much about the complex, this is a megadalton-sized complex, and it localizes in the inner membrane and concentrates at these cristae junctions, the site of invagination of the cristae membranes. So this is an amazing complex because if you get rid of its components, you drastically affect mitochondrial architecture and you drastically affect numerous different mitochondrial functions. The other thing that's amazing about MECOS is not only is it affecting the shape and organization of these cristae membranes by its behavior at cristae junctions, but it also has a number of different interactions across mitochondria. So all of these arrows here are different interactions between the MECOS complex and other complexes on the outer membrane. And these interactions can be very tight to the point of which there's a super complex between MECOS on the inner membrane and the SAM complex, which is involved in beta barrel assembly on the mitochondrial outer membrane that's present in human cells. MECOS is also interacting with the respiratory complexes and the mitochondrial fission and cristae modulating machine, the OPA1 machinery on the mitochondrial inner membrane. So to some degree, MECOS has its fingers in all of the pies of organization of the inside of mitochondria. And what I think is amazing about it is that the protein complex is essentially completely conserved from budding yeast all the way up to human cells. And because of this, we can study different behaviors of MECOS in different model organisms and learn a lot about this various aspects of mitochondrial organization. So these are some images that we've taken in our lab. This is in budding yeast or in humans. And if you label MECOS proteins with GFP, or if you use immunofluorescence in humans, what you can see is that it takes on this semi-punctate dotted nature. And wherever you see these dots, we think this corresponds to where these cristae invaginations are on the mitochondria. And so on top of being very important for cristae organization, they're also just quite beautiful to look at. And for someone who loves microscopy and loves thinking about spatial organization, I think that presents a number of really interesting questions and challenges. And so just to give you a couple minutes of flavor of different ways that we've been trying to address these different questions, I'm going to highlight a couple of the studies that we've been working on the last couple of years before I get into our interest in mitophagy. So one of the first things that we've been able to figure out, really honing in on budding yeast as this model system, is that we've been able to figure out that MECOS is organized into these two major subcomplexes. And what we've figured out is that if we look at where they localize within the mitochondrial network, here is a MECOS subunit labeled in purple, the mitochondrial network's in blue. What you can see is that this dot inside the mitochondria at the inner membrane is really corresponding nicely with contacts with the endoplasmic reticulum. And this is budding yeast ER labeled in green. And you can see as the ER is moving around, this MECOS protein on the inner membrane is seeming to respond to these interorganelle contacts. And this is just one example of how we think these interorganelle contacts are really tuning into the internal organization of mitochondria. So for some part of the lab, we're really interested in these cristae organization and MECOS. But what we found is by looking at MECOS, we've really stumbled onto these mitochondrial dynamics pathways. So we started off by doing the screen in budding yeast, looking for factors that affect MECOS localization. And we stumbled across this protein that sits inside mitochondria and is actually required for the fission of the organelle. And so there's been this question that's been present in the dynamics field for probably 20 years, which is, how do you coordinate mitochondrial division across two membrane bilayers? And what we think is that we've identified at least the first thing that we know is required. This is a budding yeast protein that isn't necessarily conserved in humans, but it suggests that there are these internal factors that are cross-talking with both MECOS and also with the dynamics of the organelle that is really kind of tuned into these interorganelle contact sites. And then lastly, I wanted to highlight one of the newer studies that we have in the lab that I'm not going to be going into in detail today, but I wanted to point out that we're using fission yeast as another model system because it has a really interesting pared down version of the MECOS complex with only a few core subunits. And by looking at this, we uncovered was that there's an additional protein that's present in most fungi, just happens to be missing in budding yeast and was kind of missed along the way. But what we think we found is that there's this MECOS interacting protein that scaffolds the cristae membrane from the matrix side of the membrane. And so I'm not going to get into more detail on it today, but I wanted to highlight that my postdoc, Abhishek Kumar, who really has made leaps and bounds in terms of our understanding of this process, is going to be giving a talk in a different online seminar series, the Pombe Talks, tomorrow. And it's at the same time as this one, and I highly recommend you check it out if you want to learn more about kind of weird things about MECOS that seem to be present in fungi. Okay, so I want to change gears a bit and kind of talk to you about what you're all interested in, which is mitochondrial quality control. And And from our perspective, we're really interested in the context of these different things, how quality control is spatially organized in the mitochondria. And like many of the projects that are going on in the lab, we kind of stumbled onto this a little bit by accident, by looking at these proteins that seem to be interacting with mucose, but not, we think of them more as auxiliary components of the complex. And so I want to bring back this cartoon that I really like. This is done by Klaus Bonner's group and people in his group. And I want to highlight this protein that's localized to the mitochondrial intermembrane, this protein TMEM11. And basically, we got interested in it because if you look at reviews about mucose over the years, it's always drawn with this arrow on the intermembrane with kind of a question mark. And the reason that it's drawn on these mucose reviews is in part from two different studies. So one is work by Wade Harper's group, which did IP and mass spec of different mucose components. And they always saw that TMEM11 showed up as one of these common interacting partners. And the other reason that we were interested in TMEM11 was work that was done in flies by Julien Royer and Thomas Rival, where they looked at the TMEM11 homolog in Drosophila, so the protein called PMI. And they characterized the protein as a mitochondrial intermembrane protein. And the knockout of this protein leads to defects in cristae organization. You can see that the cristae in these mitochondria look highly aberrant, very elongated and curved. And then secondarily, that the mitochondria in these knockout cells balloon up in size. So this normal mitochondria on the left is drawn to scale with these giant mitochondria that they observed in the absence of PMI in flies. And so because of its localization of the intermembrane, its interaction in with mucose, and its defects in cristae, we were really interested in this protein and wanted to know what it might be doing and how it could work with mucose to maybe fine-tune cristae organization. Now, beyond that, when we started the study, not much else was known about TMEM11. It's a polytopic membrane protein with 3 transmembrane domains. It has no known functional domains, but that didn't really deter us. And so when our first student joined the lab, this guy Oz Gok, who's now a 6-year student in the lab, basically we set to work to ask, well, what's the role of TMEM11, and what might interesting things could it be doing with mucose? And so when Oz joined the lab, the first thing he did was he made these stable knockdown lines using CRISPR-I and looked at mitochondrial morphology. And what he saw was basically the exact same thing that was previously observed in flies, that if you get rid of TMEM11 with 2 different guide RNAs, you can see that the mitochondria balloon up in size and have this really aberrant structure. We did EM of these mitochondria, and again, the images on the left of our control cells are drawn to scale with the knockdown cells. You can see that the mitochondria really swell up and have this ballooned phenotype, but also that just like was reported in flies, that the cristae seem to be very long and very curved. And going into this project, we weren't really thinking that TMEM11 could be having a non-canonical role. We really thought it would be doing something with a mucose complex. And so, we looked at this as a cristae defect. I think kind of now what we know now, I think that this is more of a secondary phenotype, but I'll tell you a bit of the story about our initial studies of this protein and then how we got interested in mitophagy. So when we started the project, we were really interested in studying whether mucose defects could be causing this mitochondrial morphology defect. And I know many of you probably are familiar with the technique of blue native page and these types of gels. So I'll go through this really quick for those of you who may be less familiar with this. This is really a great assay for looking at protein complex sizes, particularly within mitochondria. And it's known, I mentioned before, that mucose is assembled into a megadalton size complex. And these kinds of assays are one of the ways that this was figured out. So the way this works is you can purify mitochondria. You can use a mild detergent like digitonin to separate the organelle to protein complexes. And then you can run the samples on a native page gel. And then for a 2D gel, which is what we do here, what we do is we cut out the band, turn the gel on its side, and then run it in a second dimension with STS page. And then you can probe for your favorite protein of interest. And so here's one of the subunits of the mucose complex, which runs at about 27 kilodaltons in size. And to orient you on gels like this, the right side of all these gels that I'm going to show you are low molecular weight complexes, and then the left side of the gel are all the high molecular weight complexes. So what we did was asked whether or not TMEM11 loss was affecting mucose assembly. And so here I'm showing you a different mucose subunit, MIC60, and then we looked in either control cells or cells where we've depleted TMEM11. We purified mitochondria from each and then ran these gels. And I'm going to show you quantifications of these where the black lines are essentially looking at the fluorescence or the intensity profile of these Western blots. The red would be characterizing our TMEM11 knockdowns. And what you can see is that the profile for this is largely the same whether or not you have TMEM11. Again, when we started this, we really had a mucose-centric view of what was going on. So what Oz did was he looked at every single mucose complex member and could not see a defect. We also considered maybe that TMEM11 is some sort of auxiliary assembly factor for mucose, but really could find no evidence for that. So what we instead did was we started looking at TMEM11 itself and asked whether or not it was part of mucose. And what Oz saw when he did lots of TMEM11 was that most of the protein was localized in much smaller molecular weight complexes, but had this range from the bottom of the gel all the way up to about 500 kilodaltons in size. But then secondarily, we also saw large molecular weight complexes, just not as intensely. And then we looked in our cell line that we generated where MIC60 was knocked down, and you can see that TMEM11 was basically unchanged at these lower molecular weight sizes. But I want you to notice that also there are these high molecular weight complexes that seem to be mucose dependent for TMEM11 to associate with. And I'm going to come back to that point at the very end of the talk. So the question that then became, that we wanted to ask is, okay, well, TMEM11 is seemingly not required for mucose assembly, but maybe it's going to the same place as mucose to do its function. And so we wanted to ask where TMEM11 localized within mitochondria. Now, when we started this project, the antibodies that existed for the protein weren't particularly good. And so what Oz did was he generated tagged versions of TMEM11 that we reintroduced in our knockdown lines. And there are two different lines that I'm going to be talking about. One where we tagged TMEM11 with GFP, and then another where we tagged TMEM11 with both Apex and GFP tags. And what I want you to notice is that the mitochondrial morphology is completely fixed when we introduce TMEM11 into these cells. And this is really where we got our first surprise in the project. We used this Apex tag to do proximity labeling and see where TMEM11 localizes within the mitochondrial network. And what we saw was that we were kind of expecting when we started this that TMEM11 would concentrate at the cristae junction on the mitochondrial inner membrane. And what Oz instead saw was that our precipitate, when we add diaminobenzidine, hydrogen peroxide, was actually occurring on the outside of the mitochondrial outer membrane. And so this was in contrast to what had been previously reported from that fly work. So what we did to confirm this was we turned back to a much more traditional technique. We use a protease K protection assay where we purify mitochondria and then add protease K either before or after disruption of the mitochondrial outer membrane. And what I want you to notice is that MIC60 here is not affected by adding protease K unless we also disrupt the outer membrane. And instead, TMEM11 was getting degraded immediately upon addition to protease K, just like an outer membrane protein like TOM20. So we revised the topology of it. And what we know is that TMEM11 is likely facing the cytosol with its N-terminus, and that's based on where the Apex labeling was giving this precipitate. I mentioned before that it's a polytopic transmembrane protein, and in fact, very little of the protein is facing the intermembrane space where mucoses would be facing. So what we wanted to do is then use the tools that we generated to ask, what's TMEM11 interacting with in the outer membrane, and could that be giving us a clue to why we're getting these morphology defects? So what I'm showing you here are duplicate experiments where we did IP mass spec of GFP tag TMEM11. On one axis is a single replicate, on the other axis is another, and anything along the diagonal is reproducibly coming down in both times we did this experiment. And what you can see is that this worked really nicely. TMEM11 jumps to the top of the page. And what we also noticed was that we could really nicely recapitulate what Wade Harper's group has shown, which is that TMEM11 is interacting with these mucose subunits. So all of the core mucose subunits are highlighted in blue. I mentioned earlier in the talk that mucose forms this super complex with outer membrane proteins. Those are highlighted in magenta here. So all of those really came down nicely with TMEM11. But what we also saw is other abundant outer membrane proteins, the VDACs. And then the reason that I'm here talking to you guys today is that we also noticed this interaction with BNIP3L, which is also called NIX, and BNIP3. And as I'm sure many or all of you are aware, these are mitophagy receptors that are tail anchored in the mitochondrial outer membrane, and they have LC3 interacting motifs that face the cytosol and are required for recruitment or allow recruitment of LC3 to promote autophagosomal membranes that surround mitochondria and target them for degradation. Now, I have to admit that, mitochondria are amazing, and there's so many subfields within the study of the organelle that even for someone like me who's really interested in internal organization, I obviously was aware of the work that Richard Yule started with Pink-1 and Parkin, and that, the interest in the field in mitophagy. And I feel like I was a little bit less aware of this receptor-mediated mitophagy pathway. And this is in part because I think when we started this work, people primarily were thinking about BNIP3 and NIX in terms of the kind of specialty pathways that they're really implicated in. So for starters, these are proteins that are known to be targeted transcriptionally by HIF1 alpha, and under hypoxia, they're known to be massively upregulated to promote mitophagy. And then secondarily, NIX is a protein that's implicated in development, and particularly it's involved in the clearance of mitochondria from red blood cells during erythrocyte maturation. So the fact that we were seeing it in our IP mass spec of a cell that wasn't really treated with any sort of stress was kind of a surprise, at least to me. And so before we kind of jumped whole hog into looking at mitophagy, we wanted some additional evidence that BNIP3 and NIX were worth studying. And so what we did was we turned back to our 2D blue native page gels for any sort of evidence that they might be actually either interacting with or kind of playing a role with TMM11. And so what Oz did was he went back, he did 2D blue natives. We saw that BNIP3 as well as NIX were kind of in a similar size complex to TMM11. So that was kind of a point in favor. But I want to point out that if you do these gels, and I think anybody in the audience who's familiar with this knows that a lot of proteins will actually have this kind of smeared pattern or size similarly to these complexes. And so the VDACs are a great example of this, that they run at almost an identical molecular weight to TMM11. And so Oz had this really good idea. So he reasoned that we'd put these huge tags on TMM11. We're talking about a 20 kilodalton size protein that's had 60 kilodaltons worth of tags fused to this N-terminus. And so Oz reasoned that if we put these big tags on TMM11, we could see a shift when we ran these 2D blue native page shells. And that's exactly what we saw. So TMM11 normally runs up to about 500, although we get a bit of a smear at high molecular weights. But then when we put the tags on TMM11, we saw this leftward shift in the gels, and this is just quantified here. The red that I'll show you here is the Apex and GFP tag TMM11 compared to the black. And then what we did was we used those purified mitochondria to ask if there was an effect on BNIP3 and NIX, and there absolutely was. In both cases, you can see this leftward shift on these gels and the high molecular weight movement of them into these complexes. And then what was really satisfying is when we looked at VDAC, even though it was abundantly pulled down in our IP mass spec, it wasn't undergoing this shift in these gels, that it stayed at exactly the same size. So to us, this really indicated that there's specificity between the interactions between TMM11 and BNIP and NIX. The other thing that really got us clued into these proteins playing a role together was when we asked whether or not TMM11 mitochondrial morphology defects actually required these BNIP3 and NIX proteins. Again, when we started this project, we really were thinking about it from a mitochondrial morphology centric standpoint. And so what Oz did was he took our control cells and introduced siRNAs, either a scrambled control or siRNAs targeting BNIP3 or NIX. I'm showing you just BNIP3 here. You can see that when we knock down BNIP3, we really don't have drastic changes in mitochondrial morphology. But when we introduced BNIP3 siRNA into our TMM11 stable knockdown cells, what we saw is that we could almost completely fix the mitochondrial morphology defect of these cells. They looked almost completely normal again. Now, since we're here, I want to point out one other thing that I think is really interesting, and we can kind of come back to this point at the end of the talk. So I think for those of you who look at mitochondrial morphology, many of you probably noticed that even when cells look healthy as a population, there's always a few cells in our tissue culture dishes that look a little weird. They'll have a bit of a bloated behavior, or the mitochondria look a little fragmented. And what was noticed was that in our BNEP3 knockdown cells, we almost completely fixed that phenotype, and the mitochondria look beautiful. And so I think there probably is a component of this mitochondrial swelling that's associated with BNEP3's activity in mitophagy, and we can talk about that a little bit more at the end. So what I showed so far is that TMEM11 is sitting on the mitochondrial outer membrane. It does seem to interact with the mucose complex, but really what was compelling is this tight interaction that TMEM11 seems to have with both BNEP3 and NIX, and that if you get rid of TMEM11, you get this mitochondrial morphology change that seems to be dependent on BNEP3 and NIX. Now, at this point, we were convinced that we really needed to start looking at mitophagy. And there are these key differences between the PINK1 Parkin mitophagy pathway and the receptor-mediated mitophagy pathway that I think many of you are aware of, but really struck us from as biologists who are interested in the spatial organization of mitochondria. So baked into the PINK1 pathway is that PINK1 gets imported into healthy mitochondria where it gets targeted for degradation. And then, at least pharmacologically, when we depolarize mitochondria, that PINK1 accumulates on the mitochondrial surface and leads to this cascade of events that allows for mitochondria to get targeted for mitophagy. And also that this is coupled to the fragmentation of mitochondria into tiny little pieces. So what was obvious to me is that there's kind of a spatial aspect baked into this system. And the question that I was really interested in is, for this receptor-mediated mitophagy pathway where BNEP3 and NIX are tail anchored in the mitochondrial outer membrane, even though these proteins are really implicated under hypoxic conditions or a stress response role, that there had to be some way that their activity has to be kept in check. And then secondarily, there has to be something that actually dictates where they get activated within the mitochondrial network, right? How do you actually define where mitophagy is happening when these proteins are around at low levels? And so with that in mind, we asked whether or not TMEM11 could be playing a role in this mitophagy pathway. So I'm sure almost all of you are familiar with the mito-M-chyma assay that's used to monitor mitophagy. Basically, that mitochondria that are healthy are of a particular pH, and that when mitochondria are engulfed and incorporated in a lysosome in that acidic compartment, the acidic environment allows for a change in the excitation of that fluorophore and distinction between normal mitochondria and acidified mitochondria. Now I'm going to be showing images of this where all of this is pseudo-colored, and everywhere I show you a purple mitochondria that's normal, and then the defective acidified mitochondria that are in the lysosome will be colored green. And I think a lot of people who use this mitophagy assay do this at a population level, and they'll use a FACS-based system looking at the fluorescence ratios of these different excitation of mito-M-chyma. And what we noticed when we tried this assay is that the effects that we were seeing in TMEM11 knockdown cells, particularly in normoxic conditions, were not really, they were subtle enough to not be detected by that assay. And so what Oz instead did was he went around and painstakingly counted up all of the different cells and how often he saw one or two or more punctin cells. And so what I'm showing you on the right here are plots where he's actually indicating how much mitophagy he sees in terms of puncta per cell. And so on average, this is using HeLa M-chyma cells that we got from Richard Yule's lab. He see about 1 puncta per cell, but often maybe a quarter of cells had 5 or more puncta. So there is some amount of basal mitophagy that he was able to detect in these cells. And we know that this is basal mitophagy because we were able to completely get rid of almost all of this mitophagy by depleting BNEP3 and NIX from these cells. Now I'll point out, and I'm sure you all are aware, that these cells are not expressing Parkin. We did this experiment again in U2OS cells, which is kind of one of our favorite models in the lab. And I want to point out, I'm not showing the data for this, but this basal mitophagy does not seem to be alleviated by BNIP3 and NIX depletion. It's possible that there's interplay between the different mitophagy pathways that are going on. But the common theme in both systems seems to be that when we deplete TMM11, we get this marked increase in mitophagy. So what I'm showing you here is an image where we get mitochondrial swelling in these HeLa cells in the absence of TMM11, but also a really drastic increase in how many of these puncta that we see. And again, that increase is completely dependent here on BNIP3 and NIX. If we get rid of all three proteins, the amount of mitophagy we get in these cells goes back to nearly zero. Now, these data are statistically significant, but again, as we kind of stumbled around into our interest in mitophagy, we weren't quite convinced at the beginning that this was a meaningful result. And what we wanted to do was take advantage of the fact that it's known that BNIP3 and NIX can really become stimulated upon hypoxia. So I mentioned at the beginning that these are both proteins that are stimulated by HIF1 alpha, and they're transcriptionally upregulated. And you can see that in these HeLa cells, but also in these U2S cells, you get a massive increase in how much BNIP3 levels there are, as well as an increase in NIX levels. Importantly, TMM11 does not seem to be affected in its protein levels by treating with a drug called cobalt chloride, which we used that actually mimics hypoxia. I also want to point out that we've now gone back and done this with other sorts of treatments like DFO, which stimulates iron chelation-mediated mitophagy, and we've also started looking in hypoxia as well. And in all these cases, TMM11 seems to be having similar effects. So now when we treat with cobalt chloride for a day, instead of getting 1 or 2 puncta per cell, we're getting massive amounts of puncta. On average, about 20, but you can see cells with 80 or even more puncta per cell. And what was really satisfying is if we got rid of BNIP3 and NIX again, we almost eliminate this mitophagy. And then what was extra satisfying was that if we knock down TMM11, you can see again the mitochondrial morphology effect becomes even more pronounced in these cobalt chloride treated cells, and then just these cells with massive amounts of mitophagy going on. So an increase from about 20 on average to 30 or even more, but you can see this, this population here at the top that really expands in terms of massive amounts of mitophagy. And then again, when we get rid of BNIP3 and NIX altogether, we almost completely eliminate the mitophagy that's going on. Now, one of the things I want to point out is that if you deplete BNIP3 and NIX under cobalt chloride treatment, the cells will actually die unless we simultaneously treat with a caspase inhibitor to prevent apoptosis. And so, the mitochondria here, they don't look quite normal, but they become tubular in nature. We both fix this swelling defect and then again prevent this mitophagy. So at this point, we saw that TMM11 is in this complex with BNIP3 and NIX. And if you stimulate with cobalt chloride and you don't have TMM11, you both get increased mitophagy, but also this mitochondrial swelling phenotype. And our naive model at this point was that what TMM11 could be doing was actually forming this complex to keep BNIP3 and NIX activity in check and actually preventing them from inducing mitophagy pathway. And the prediction that would arise from that model is that TMM11 would localize with BNIP3 and NIX before mitophagy induction, and then you'd have a release of BNIP3 and NIX when you initiated mitophagy, for example, when we treated with cobalt chloride. And so what we did to start looking at this was to transfect in GFP tag TMM11 into our cells, and then we would fix and label mitochondria with a general marker like TOM20, as well as BNIP3. And the first half of that model seemed like it was going to be true. What we noticed was that TMM11 was kind of widely distributed on the mitochondrial network, but it would enrich in these discrete foci and seemingly co-localize often with BNIP3 and the foci that it would localize to within the mitochondrial membrane. But the thing that shot the model out of the water was when we started treating cells with cobalt chloride. And what we saw was that BNIP3 would redistribute within the mitochondrial network. You can see its localization here within mitochondria, but also it really discretely enriched into these large punctate structures. And what we also saw that was really exciting to us was that Team M11, instead of releasing these proteins, seemed to be going where Team M11 was going. Now, it struck us that these circular structures look a lot like the ring-shaped structures that you see when you label autophagosomes, particularly enlarged ones. And so we didn't think that this was a sequestration event, but to rule this out, what we wanted to ask is whether these foci were indeed mitophagy sites. And so what Oz did was he co-transfected cells with Team M11 as well as LC3, and then he'd fix and stain for BNIP3. And what we saw was that almost every time we saw a BNIP3 foci, Team M11 would be there about 3/4 of the time, and then 95% of the time, a BNIP3 foci would be co-localized with LC3. Now, one of the things that we've gotten interested in since we've published this work is really examining the coupled dynamics of these proteins. And so I'm going to show you a video where we've now transfected cells with cherry tag BNIP3, as well as GFP tag Team M11, and then BFP tagged LC3. And then on the left is just the mitochondrial appearance. And so all of these are synchronized. And when I play them, what you'll see is at the site of this arrow, that BNIP3 is going to accumulate at this spot within the mitochondrial network. So, I mean, going back to what I mentioned before about CCP-induced mitophagy with Pinkhorn and Parkin, and that you get this fragmentation of the mitochondrial network, we think we're seeing something slightly different here, where BNIP3 and Team M11 and LC3 are all simultaneously kind of accumulating at this discrete site within an intact mitochondrial network. And really, I think indicates that there's something spatially coupled about how these proteins behave. And so what we've seen is that they dynamically accumulate, and that, again, these are at intact mitochondrial tubules. So to kind of update the model, what I've shown you so far is that Team M11 is co-localizing with BNIP3 and NIX, and that you get this low amount of basal mitophagy, at least in HeLa cells that are dependent on this pathway. And then if we do a treatment, for example, with hypoxia, you get extensively increased levels of BNIP3 and NIX and much more mitophagy on the mitochondrial network. And what I showed is that Team M11 is going to the site regardless of whether or not there's basal mitophagy or this hypoxia mimetic induced mitophagy. And so we started thinking about how could we update the model, and what could be another role of Team M11? Why would it be going to these mitophagy sites, but that we'd get more mitophagy without Team M11? And what we're wondering is maybe what's going on is that Team M11 is there to help dictate the site of where mitophagy is occurring, right? If you have BNIP3 and NIX and they're distributed through the mitochondrial network, what helps decide that that site in the movie that I just showed you is the site where mitophagy is occurring? And so what I showed is that we get more mitophagy without Team M11, and we wondered if maybe what Team M11 is doing is helping limit and dictate those sites. And so the prediction that would come from that model is that you'd get some sort of indiscriminate mitophagy and much more mitophagy spatially without Team M11. And so the way that we kind of started to test that model was to look at where BNIP3 localizes within the mitochondrial network during mitophagy in the presence and absence of Team M11. And what we saw was that when we knocked down Team M11 here on the bottom, we'd get these cells with just drastic numbers of BNIP3 sites. So I showed you using the M-chyma assay that we get lots more mitophagy in these cells, but in fact, we're also seeing simultaneously stimulation at many places within the mitochondrial network of active mitophagy sites. So instead of seeing this maybe in one place at a cell at any given time, now we're seeing cells with 5, 6, even more than 10 mitophagy sites at once. And so our model is that what Team M11 might be doing is kind of interacting with NIX in this complex and performing this surveillance of the mitochondrial outer membrane and waiting for a signal that says that there is a stress or some sort of dysfunction that needs to initiate mitophagy. And so when you have something that is sensed by the mitochondria, like hypoxia, that you get this local enrichment and accumulation of BNIP3 and NIX at these mitophagy sites. And that what Team M11 might be doing is helping spatially define where that mitophagy occurs. Now, the beginning of the talk, I said what the focus of the lab is really interested in this internal organization. And so we really were kind of taking this mindset of wondering why Team M11 is interacting to small degree with the MECOS complex, and why you have a little bit of those high molecular weight complexes of Team M11 that seem to depend on MECOS. And from the spatial regulation model, one of the things that we wondered is MECOS is really primed at the mitochondrial intermembrane by its localization at individual cristae junctions to sense whether there's dysfunction locally within that mitochondrial network. And so what we're wondering is whether Team M11 might be interacting with MECOS as a way to trigger and locally induce that mitophagy. Now, this is a really hard question to get at without knowing the specifics about how Team M11 interacts with MECOS. That's something that we're actively working on in the lab. But I want to show one piece of data that suggests that maybe that model might be right. And so what we did was we got rid of the MECOS complex by destabilizing that core member, MYC60. And what we did was we looked at how much basal mitophagy is going on in these HeLa cells. And what we saw is that if we siRNA treat with MYC60, we got an induction of mitophagy to a very similar extent as if we got rid of Team M11. And really importantly, getting rid of MECOS seems to stimulate BNIP3 and NIX-mediated mitophagy, because if we simultaneously knock down all three of these, the basal mitophagy goes back down to near negligible levels. And so this doesn't prove that this model is true, but it does suggest that there's this, this way that cells can sense MECOS dysfunction and trigger this mitophagy pathway. So I want to wrap up there by highlighting the people that did the work. I mentioned Oz before. He's the student over here and really did a phenomenal job for a lab that's doesn't have much expertise in mitophagy, and really took us in this new direction. And so he did a phenomenal job on this project. And Olivia Connor, who's a technician in the lab, helped him out with this, some of the assays in this paper. I also want to point out that we have a really fantastic environment at UT Southwestern, and we did some of the work characterizing our knockdown cells in collaboration with Sean Mishra, who's here. I want to thank Richard Yule for really generously sharing the chyma cells and kind of getting us off the ground and doing some of these mitophagy assays. And then lastly, I want to point out that we have a collaboration that we started with Natalie Nemi at WashU, and she's interested in other factors that seem to be regulating this receptor-mediated mitophagy pathway, and has a new story out in NatCom that you should be sure to check out. And with that, I'll stop there, and I'm really happy to take any questions you might have.
Well, thank you, Jonathan. That was a really beautiful talk. I would invite people still to submit questions if they have. The first ones are coming in, so I guess we get started with those. And the first one would be from Erica Holzbauer. She says, beautiful talk. I'd love to hear more about your live cell experiments and the relative kinetics of recruitment of BNIP, TMM, and LC3.
Yeah, so we did a little bit of that experiment, and basically what we've seen is that they seem to all show up within the same time in the context of those experiments. Now, the thing that we found when we did that is that, and many of the people in the field know, is that if you overexpress these mitophagy receptors, you'll actually drastically change how the system works. So BNIP3 overexpression or NIX overexpression will cause massive amounts of mitochondrial fragmentation, induced mitophagy. And so we had to be really careful in those experiments to not conclude too much. What we've really gotten interested in doing is finding ways to label the endogenous protein. And so one of the things that we're actively working on is making lines where we can actually visualize the endogenous mitophagy pathway induction, and hopefully, get more precise answers to those kinds of questions.
Then there's a question from Anson Lee. Thank you for your talk. Do you know if the DRP or mitofusin fission fusion pathways were affected upon TMM knockdown?
Right. We haven't looked too carefully at that. One of the things that we've kind of noticed along the way is that there does seem to be a little bit more of a fused network in our Team M11 knockdown cells. But that's absolutely an interesting question of how these morphology changes are coupled to mitophagy pathway. One of the things that I didn't highlight at the end that we're really interested in is, why do you have this change in mitochondria and the swelling that's induced by Team M11 knockdown, and why is it BNIP3 and NIX dependent? And so one of the interesting things that we're kind of looking to pursue is we know that during hypoxia treatment, you get this mitochondrial swelling. And it's been shown, at least in certain cell types, that that is also dependent on BNF3 and NIX. And so I think that there's got to be this interplay between shape change and mitophagy induction. And so we don't know yet if it's a good thing for the cells and that you get maybe protection of parts of the mitochondrial network by having that change shape, or if it is it's a kind of a sign that things are damaged. One of the experiments that we did was we found using seahorse assays that even though the mitochondria look really drastically abnormal in the absence of TMEM11, that functionally they're perfectly fine. That the cells grow well, and by a seahorse assay, the mitochondria are capable of respiration. So I think going in, we thought, oh, the mitochondria look bad, they must be not happy, and that might not actually be what turns out to be true.
Okay, I mean, it's quite amazing what these organelles are capable of. Next question's from Juliet Goldsmith. She says, thank you so much for this great talk. Could you comment on the role of BNP and NIX in remodeling fold development and potentially the relationship between mucose regulating glycolysis versus oxos?
Okay, so the first question about BNP3 and NIX during development, I mean, there are certainly, as I said, like during red blood cell development, that is NIX's job. I think the question that, what we're interested in is whether TMEM11 might be playing this job in spatial regulation in certain pathways and not in others. So if you think about wanting indiscriminate mitophagy, where you want complete removal of mitochondria, perhaps TMEM11 would not be necessarily involved in that process. Or perhaps, that's something that as development occurs, that TMEM11 might become decoupled from mucose. I think, there's nearly limitless questions in different tissues that would be really fascinating to look at and ask these questions. As far as how mucose remodels mitochondria during changes in oxphos, we know that, like many mitochondrial proteins, you get more protein levels of mucose that corresponds with the increase in cristae density. I think it's a really interesting question of how you get that remodeling. And certainly we're interested in understanding that. I think, mucose is always drawn as this simple, discrete complex that's at cristae junctions. And I think what's becoming apparent to us is that, yes, that can be true, but mucose is doing many more jobs, and there are certainly subcomplexes, and it's not just one discrete thing. And I think, the angle that our lab is trying to take is understanding all of these unique roles. And I think it goes at asking all these questions about all these peripheral factors that interact with mucose and what jobs they might be having. Here we found that clearly there's a protein that's linking that complex to mitophagy to some degree. But as I went back to the beginning of the talk about spatial organization, all of the different functions have to have this organization. And I think mucose is like a great way that mitochondria can kind of take advantage of that, because it's so sticky and it can have all of these different protein interactions.
The next question is from Valentina Cianfanelli. Very nice talk. Maybe I missed it, but I was wondering whether the iron chelator induced mitophagy is also affected by TMEM11 and its functional interaction with BNP3 and NIX.
Yeah, so we didn't publish any of this in our work. We started looking a little bit now. It seems to be that we get a similar increase in mitophagy when we treat with iron chelation, but I don't have that in any sort of quantitative way to show.
And maybe just me chipping in there, I saw that in the plot that you showed, the TMEM11 levels seem to stay the same, right? They do not increase. So would that mean that there's always sort of an excess of TMEM11 for increasing levels of NIX and BNP? Yeah,
that's a great question. I mean, the immunofluorescence that I showed with the GFP tagged TMEM11, it seemed to be more distributed. We've since gotten a decent working antibody with TMEM11, and it looks a little less everywhere and more at discrete punctae. But it's really hard to tell because you get so much background immunofluorescence signal that it's hard to tell if the dot you're looking at is real or it just happens to be there's background signal at that spot. So it's a great question what the relative stoichiometry of TMEM11 and BNP3 and NIX are. We just don't have a good answer to that. But the prediction would be that it's there in excess to begin with, and then, and so yeah, that's a good question. Okay. There's plenty of questions, so I may just have to take a few more because I see we're
reaching the end of our time. The next one will be from Lucas Restrepo. Interesting talk and beautiful microscopy. I'm wondering if you see any changes in the mitochondrial DNA nucleoids with TMEM mutants. I know that MECOS can affect CRISPR mitoDNA. And a second related question would be, have you looked at whether PINK1 parkin mediated mitophagy is affected by TMEM11?
Right, that's both great questions. So as far as the mitoDNA distribution, it seems like they are kind of accumulating in these fat part of the swollen parts of the mitochondria. And that's maybe why we're thinking that the mitochondrial swelling could be this protective response where you don't necessarily get mitophagy at those sites. Maybe the thinner parts of the mitochondria are where the mitophagy is activated. We really want to start addressing that question and ask, if we had a way to uncouple the mitochondrial swelling from the induction of mitophagy, we might be able to ask, why do you have that swelling, and whether this is a good or a bad thing for maybe protection of things like mitochondrial DNA. You'll have to remind me that, you were asking about pink parkin mitophagy. We haven't yet looked. What we can say is that in U2OS cells, where at least there's small amounts of pink parkin, we know that the basal mitophagy there doesn't get stopped by depletion of BNF3 and NIX. But we also know that if you get rid of TMEM11, you get an increase in BNF3 and NIX-mediated mitophagy. So what that tells us is that other mitophagy pathways there are active, and that the effect of TMEM11 seems to be limited to the BNF3 and NIX pathway. And that's consistent with what our protein-protein interactions are showing. It's certainly possible that there are crosstalk between those pathways. And I think that's another question that I think the field really needs to get at is all these different mitophagy pathways, and how do they talk to each other? How do they know which one's going to be active, particularly in cells and tissue types where you have more than one thing capable at a time of inducing mitophagy?
And then maybe one last question before we round up. I'll try to fuse a few of the questions that came up. There are some questions about the mitochondrial swelling that you see in these clusters of TMEM and NIX-BNF. And basically the question centered around, is there anything special about these clusters of TMEM, NIX-BNF in terms of mitochondrial DNA, maybe the ER in the vicinity? Is there anything that you've looked at so far?
Yeah, all of great questions. So we want to look at the ER. We would love to look at the relative localization of these proteins. I think this is, going back to Erica's question at the beginning, there's fascination now of the dynamics of the system. And I think what's really disheartening is that you can't just look at BNF3 in live cells without screwing up the system. And that's kind of why we're really focused on now trying to study the endogenous pathways, so we haven't altered these types of things. And I think that's an area that, given our interest in microscopy and some of the skills that we have in the lab, that perhaps we have a particularly good angle of being able to try and address these questions.
Well, thank you, Jonathan. Still a few more questions in there that we just don't have the time to go over. I'm sure that if people still have burning questions, they can email Jonathan, and he's going to be very happy to reply to those. Yeah. Then all that's left for me to do is announce our next speaker. So we've hit our Christmas break now, and we'll continue again in January with Angelica Haubauer, who I invite everyone to join. She had a really beautiful paper just out. So I'm sure she's going to tell us more about this and perhaps some other work of her. So I wish all of you a very nice Christmas break, and then I hope to see you again in the new year. Thank you.
Yeah, thank you, everyone. And thank you, Jonathan, for joining us.
Thank you very much for having me.
I think we can spare the coming session and also save you time. I think this was a very lively discussion.
Okay, well, I'm happy to stick around more, but up to you.
I think I got some feedbacks from the team that not many people would be able to connect, so they were a bit of conflicting. So I saw some of them have asked you questions within this session, so I think it would be all good. Thank you, Jonathan. Then we stay in touch.
All right, definitely.
Have a good break.